projector is doing something weird. It doesn't seem to be the adapter, but the color is kind of coming out weird, so we're just going to have to suffer on this together, I think. About all I can do is change out the adapter. I'm not sure, it must be something with the projector. But this background is supposed to be white, so I'm not sure what's going on. But we should be able to make it struggle together through the lecture. Okay. Okay, so today's lecture is going to be on vitamin D, calcium, and phosphorus, as well as its let me try, give me a minute, let me restart the computer, see if that actually helps. Talk amongst yourselves.
How is the brain? That's reality, man. 3 more weeks and
So today's lecture is going to be on vitamin D, calcium and phosphorus, and their biochemistry, their metabolism, and their physiological importance with relationship to bone. So the relevant reading would be in the Stepanek textbook, chapters 31 and 32. So before we start, we'll do a couple review questions from last lecture on some important concepts. So please do try to participate. You can go to this website, polo.com, NST103, or text NST103 to this number. So this first question, which of the following plays an essential role in blood coagulation? So iron, vitamin B12, heme, vitamin K, or cobalamin? Good. So vitamin K is correct. So K, think of coagulation. These other components are all important for blood. Vitamin B12, of course, the same as cobalamin, right? They're the same molecule. And then heme and iron, all of these are important for blood for different reasons, but only vitamin K is directly required for the coagulation cascade. So what is the actual biochemical function of vitamin K, and why is this important for coagulation? Because it mediates the methylation of proteins. It's required for protein carboxylation. It participates in thymidylate synthesis, which is important for DNA. It regulates nuclear receptors that control gene expression, or it monitors iron and binds 5' IRBs. Okay, so B is the correct answer, that it's required for carboxylation. We did talk about another vitamin, so vitamin B12 and also folate, right, are going to be really important for methylation. The SAM reaction, the methionine synthase, and that cycle. So those are important for methylation, as well as folate is important for thymidylate synthesis. But vitamin K coagulation is required for carboxylation. So just remember that vitamin K, what it does is it acts as a coenzyme or cofactor for this enzyme called gamma glutamyl carboxylase, or gamma carboxylase, which sits in the endoplasmic reticulum. And after translation of a secretory protein, the signal peptide is removed, and then there's a propeptide which initiates interaction with this gamma carboxylase. And then together, through its coordination of vitamin K, it's able to then carboxylate glutamate residues proximal to that propeptide sequence, right? These then can be secreted through the normal secretory pathway into the extracellular space. Okay, so the anticoagulant warfarin inhibits blood coagulation by one of these, inducing vitamin K degradation, functioning as an antagonist of factor 7, inhibiting the binding of calcium to carboxylated glutamate residues, impairing uptake of vitamin K from the GI tract, or inhibiting vitamin K epoxide reductase. majority has it. So it's through inhibiting vitamin K epoxide reductase. Okay, so if we recall this vitamin K cycle that we talked about last time, right? So vitamin K, this is just the R group, this would be a long acyl chain. The vitamin K is required in its reduced form, its most reduced form, hydroxyquinone, 4-gamma carboxylase to mediate this addition of the carboxyl group, right? And afterwards, it's converted into vitamin K epoxide. So it has to be recycled in order for this reaction to continue. So it's recycled through sequential actions of this enzyme, vitamin K epoxide reductase, back to this reduced hydroxyquinone form, right? And we can keep on carboxylating proteins. So this is the target for warfarin as well as coumarin, which is going to reduce carboxylation of proteins and thereby reduce coagulation cascade in the blood. Okay, and this is kind of a slow-acting drug, right? Because it's going to only affect newly synthesized proteins. So the proteins that are already out in the blood has no effect on those. They're already carboxylated, they're circulating. So depending on their half-life, that's why this is kind of a slow-acting drug. So it takes a while for those proteins to turn over. One more. So the intrinsic and extrinsic coagulation pathways activate this key protease, which then is responsible for directly cleaving fibrinogen. Prothrombin, factor 10, fibrin, factor 7, or intrinsic factor. Take a look. Okay, so this was incorrect. So factor 10 is definitely important. That's the convergent factor, right? Both of them converge on factor 10. So both intrinsic and extrinsic pathways will activate factor 10, which is indeed a protease. But prothrombin is actually the protease which mediates the cleavage of fibrinogen. So everybody got it correct. So this is, if you just remember the pathway, you probably don't have to remember all of this, but there are some key points that I would like you to remember. So for the intrinsic pathway, we should remember that it's activated by the exposure of negatively charged lipids. So this is going to be the key event, which triggers the intrinsic pathway. This is what would be, if you just put blood in a test tube, this is the pathway that would be activated by the negatively charged plastics in the tube. And that's why you get coagulation of the blood in the tube. So there's a proteolytic cascade. It does indeed converge with the extrinsic pathway at factor 10. So that extrinsic pathway in response to injury, you have exposure of this tissue factor, which is a glycoprotein that's usually not present in the interior of blood vessels. And this activates the extrinsic pathway, converging on activation of factor 10, which activates prothrombin to thrombin. Okay? So thrombin is what is going to be what mediates the conversion of fibrinogen to fibrin by cleavage. And then fibrin can go on to form that net-like polymer that traps blood cells and platelets and helps with the clotting process. And then thrombin, the other action it has is this feed-forward positive feedback loop. So upon activation, it goes back, it activates factor 8, and it activates factor 12, and so it feeds forward and continues to activate this pathway. Right? So it amplifies the signal. Okay, so some of the key things to remember from this are how each of these, the intrinsic and extrinsic pathway, are activated, either by exposure of negatively charged lipids or by exposure of a factor, a protein called tissue factor, that these converge on factor 10, that you guys got in the last slide. But factor 10, what it does is activates thrombin, which is a really key event in this cascade. So in addition, not shown here, thrombin also activates platelets. So it plays a really key role in the coagulation cascade. So be sure to remember thrombin and its actions on fibrin. Okay, so let's move on to today's lecture on calcium, phosphorus. So we'll first talk about calcium and phosphorus, its functions, its absorption, metabolism, vitamin D, its metabolism, and then finally bone. And kind of throughout, we'll talk about all of these compounds. So I just wanted to first start with a slide about, before we get into the nitty-gritty about the metabolism, about why we care about vitamin D, calcium, and phosphorus. And one reason is because it's really important for bone health. So in the absence of vitamin D, or if you had a deficiency in calcium or phosphorus, in babies you get rickets, or in adults you get osteomalacia, which are these bone diseases. Okay, and this is a failure of cartilage to mature and mineralize, which is essential for the formation of bone. So the actual mineralization and the deposition of the hydroxyapatite, which is going to go on to form that solid bone structure. And so you end up with this disorganization, this reduced mineralization, and this disorganization in hypertrophy, or enlarged growth of these chondrocytes, which are what produce the collagen in our joints, and which is a precursor for bone mineralization. So this gives rise to this very characteristic short stature, this widening of the long bones, and other deformations in the skeleton. So this is common in developing countries, but does also occur in developed countries as well. And so what we're going to talk about today is what is the actual biochemical function of vitamin D, calcium, and phosphorus, and why they're required for bone, and why you get something like this in their absence or when they're deficient. Okay, so what is the actual biochemical basis and mechanism? Okay, so when we think about, very basically about calcium, it has many, many functions. The plasma calcium concentration is kept at a very narrow range, around 9 to 11 mg per 100 mils. And this serves many functions. A lot of these we've touched on during the course of this class. For example, it stabilizes vitamin B12, cobalamin, intrinsic factor complexes. Particularly, calcium was required for its uptake in the GI tract because it's taken up by cubilin, which is a calcium-dependent receptor in the gastrointestinal tract, right? It's also required for the activity of many, many enzymes. We've talked about a few proteins which bind and require calcium for their activity. So in the context of blood clotting, it was actually first one of the factors, right? For blood clotting was calcium. And that's because it sits like a sandwich, bridging the interaction of the clotting factors, the carboxylated negatively charged factors, and a negatively charged lipid or molecule released from platelets, right? And calcium sits at that bridge. So calcium is required for blood clotting. It's required for this enzyme, which we've talked about, phospholipase A2. So if you throw way back to the first quarter of the semester, phospholipase A2 is the enzyme that mediates cleavage of phospholipids, right? So in the digestive tract, it's going to cleave SN2 fatty acids for releasing the lysophospholipid, which would be present in the mixed dietary micelles, or intracellularly, it cleaves the SN2 lipid for the release of arachidonic glucosinoids, right? And so that protein also coordinates the calcium, which allows it to interact with the negatively charged phosphate group on the phospholipid. Can serve important roles as secondary messengers, so downstream of G protein coupled receptors, IP3 receptors can release calcium from the ER. So in the cytoplasm, the cytosol of cells, the amount of calcium that's present is very, very low. And that's because calcium is actively sequestered in the ER, and the difference is like 1,000 to 10,000 fold. So release of calcium from the ER can be a very potent second messenger cascade. Similarly, release of calcium from sarcoplasmic reticula plays roles in muscle contraction. And then several other things we didn't really go into, but calcium functions in neurotransmission, fertility, and intracellular adhesion. Most of these are through its actions as through calcium binding proteins, because many proteins coordinate calcium. And then important for today is that calcium plays essential roles in bone mineralization, or the formation of the actual structural material which gives rise to our bones, right? And you can really appreciate this when you think about how much calcium we have in our body. So So we have about a kilogram of calcium in our body, which is a pretty huge amount of a mineral in an average 70 kilogram body weight. 99% of this calcium is in our bone. Okay? So only about 1% is present in the plasma, circulating or performing all of these other roles it plays. Right? So the bone is a major storage place and a major functional role of calcium. Okay, so similarly, the other component that we're discussing today, phosphorus, which is often present in phosphate or inorganic phosphate, serves many functions in the cell. It's present in molecules involved in energy transfer, right? So ATP, it's present in these high energy bonds in these molecules. It's a component of RNA and DNA, so we have the phosphate sugar backbone that joins nucleotides, right? It's present in many signaling molecules like phosphatidylinositol or inositol triphosphate or cyclic AMP, right? All contain phosphate. It's an important modification of proteins. So phosphorylation of enzymes or other proteins, this is mediated by kinases and phosphatases. That's a reversible modification. And it also modifies nutrients, right? It plays important roles in metabolism. Right? So probably Professor School most likely went through glucose 6-phosphate, the phosphate addition to glucose to form glucose 6-phosphate, which by hexokinase or glucokinase, right? Which is going to serve an important role in sequestering glucose in cells. It retains the glucose. It's present in the head groups of phospholipids, functions as an acid-base buffer, and it serves structural components both in our teeth as well, again, together with calcium in the mineralization of bone. And similar to calcium, the majority of phosphate in the body is present in bone. Not quite as much as calcium, but 85%, which is a lot. It's present in the bone, about 1% is present circulating in the plasma, and then the rest is present in cells serving this wide variety of functions. Okay, the majority of our calcium in terms of source is going to come from dairy products. Okay, we can also get it from tofu, from certain vegetables. And then there are a lot of supplemented foods on the market these days, right? There's like orange juice and cereals are all supplemented with calcium these days. Phosphorus is pretty ubiquitous in foods, is particularly high in protein-containing foods. So we can get phosphorus from a large variety of places in our diet. Even in like sodas and artificial foods with a lot of that are highly processed, they have a lot of phosphorus additives to them. So we're going to get a lot of phosphorus and phosphates from these types of components in our diet as well. But phosphorus is pretty ubiquitous in our diet. So we need about 1 gram per day of both calcium and phosphorus. And so intake from our diet is going to contribute to this 1 gram. And then in terms of our plasma sources of phosphorus and calcium, bone remodeling is going to be a particularly important source. You can see that in our body we have so much calcium and so much phosphorus in our bone that changes in our bone metabolism are going to have big influences on the amount of phosphorus and calcium that are circulating in the blood. Okay? So plasma calcium has to be really finely regulated. Okay, so changes in plasma phosphorus aren't as important. They don't have as significant effects as changes in calcium concentrations. But calcium has to be retained within this very narrow window, about 9 to 11 mg per 100 mils. Right? And so under conditions of low calcium, hypocalcemia, this can result in muscle spasms, tetany, and convulsions. So this would be at around 6 to 7 mg of calcium per 100 mils. And then even death, if you go lower than this, around 4 mg of calcium per 100 mils. Similarly, hypercalcemia can result in hypercalcification, which involves the calcification or the sort of aberrant deposition of calcium in soft tissues like blood vessels, like the aorta, or in organs like kidney and the heart. And this can also result in death. So calcium has to be kept in terms of plasma concentrations within this very narrow window. And so not surprisingly, our body has a lot of ways in which to regulate the plasma calcium concentrations. Okay, so in terms of absorption of calcium from and phosphorus from our GI tract, our body uses both active and passive mechanisms. So in the jejunum, and particularly in the duodenum, both calcium and phosphorus are going to be taken up. Most of this phosphorus is in the form of inorganic phosphate by active mechanisms, by active transport mechanisms. But once they reach the ileum, they're going to be taken up more by passive mechanisms. And this usually only comes into play if you have high amounts of calcium and phosphorus in the diet, where they make it past the proximal intestine to this distal portion, and then they can be taken up by these passive mechanisms. So it's going to be concentration dependent. Okay, so low amounts in your diet, you're going to take it up mostly by active. In the proximal intestine, at higher concentrations, there'll be a bigger contribution of this passive mechanism in the ileum. So you can see that there's a difference in the efficiency of absorption between calcium and phosphorus. So for calcium, about 30% is absorbed under basal conditions. However, this it's under a high amount of regulation by different hormones. So parathyroid hormone, or PTH, and we'll come back to this hormone, can indirectly increase the intestinal absorption of calcium via a metabolite, the active form of vitamin D. So we'll come back to talking about these regulatory mechanisms. Okay, whereas phosphorus, which is taken up pretty efficiently from the GI tract, doesn't undergo this really robust increase in absorption in response to parathyroid hormone. So in terms of excretion, not surprisingly, a lot of the calcium is excreted in the feces, so we don't absorb, so we absorb around 30%, so the remainder is going out in our feces if we're not absorbing that. Of the portion that is absorbed, it's going to circulate in the plasma in dynamic balance with our other stores of calcium, such as in our bone or in our cells. And about 200 mg per day is going to be excreted in our urine. Okay? And for phosphorus, it's kind of the opposite. So we do a really good job of absorbing phosphorus and phosphate from our GI tract. So we lose about 100 mg per day in the feces. About 800 of this then is lost in the urine. Okay? And so kidney reabsorption, so since this is one position in which calcium is being lost, is another potential site at which we can regulate our plasma calcium. So we can control the loss in the feces by controlling absorption, right? Or we can control the loss in the urine by controlling the reabsorption from the plasma by our kidneys. So for plasma phosphate and phosphorus concentrations, usually the reabsorption in the urine is not highly regulated. Unless it's extremely low, the amount of phosphorus excretion in the urine is going to be proportional to the concentration in the plasma. Okay? So high degree of regulation of calcium with the excretion in the urine, not much regulation of the phosphorus excretion in the urine. So there are three hormones to consider with respect to the concentrations of plasma, calcium, and phosphorus. And these are the parathyroid hormone, which I briefly alluded to, the active form of vitamin D, and then calcitonin. And this seems to be a minor contributor, but I'll come back to this. And we'll talk about the role of each of these hormones and how they regulate calcium and phosphorus. Okay, so the parathyroid gland, which is what releases the parathyroid hormone, that's going to be up by your thyroid gland or on your throat, serves as a sensor for blood calcium concentrations. So when the parathyroid gland senses that there's low calcium, it's going to increase the secretion of parathyroid hormone into the serum. And so this is shown here in this graph. Along the x-axis, we have the total concentration, total amount of calcium in the blood. And then along the y-axis, we have the amount of parathyroid hormone. So what you can see is that under conditions of low calcium in the blood, we have really high amounts of serum parathyroid hormone release, right? So low calcium releasing parathyroid hormone. Under conditions of high calcium, very little parathyroid hormone is released, okay? And this is where we want to be, right? Right around here, 9 to 11, which is the region with the most dynamic range, right, at this threshold point, right, of parathyroid hormone, okay? So low plasma calcium releasing parathyroid hormone. So the parathyroid hormone is released in response to low calcium. So not surprisingly, it's going to act and has several mechanisms to increase plasma calcium, right? So it's trying to maintain that calcium homeostasis in the plasma. So it's acting to increase plasma calcium and to some extent the concentration of phosphorus in the plasma. And so it does it through three primary mechanisms. One is that it increases the resorption of calcium by the kidney. So one place that we lose calcium is in our urine. That's going to increase the amount of calcium that's resorbed by the kidney. The other is it's going to increase resorption of bone, so bone breakdown, which is going to release calcium and phosphorus from the bone, right? So that's present in that hydroxyapatite mineral that makes up the structural components of bone. Right? So this is acting to regulate that plasma concentration of calcium. The other way is that it acts indirectly to increase the absorption of calcium from the diet. So remember that we're only absorbing under basal conditions about 30% of the calcium. So release of parathyroid hormone is going to indirectly act through a bioactive form of vitamin D to increase our absorption of calcium in the intestine. Okay? So three functions. Okay, so basically parathyroid hormone is functioning as a rheostat to control and maintain that homeostatic level of calcium in our blood. Okay? So hopefully we're all maintaining normal blood calcium levels. If there's a rise in blood calcium levels, this is going to suppress the release of parathyroid hormone by the parathyroid gland. This would result in decreased bone resorption, increased urinary loss, and decreased production of this bioactive form of vitamin D. Okay, so all of these are going to result in a decrease in plasma calcium levels, right? Decreasing parathyroid hormone. The opposite is true under conditions in which we have a decrease in blood calcium concentrations. This is going to stimulate parathyroid hormone, which is going to increase bone resorption, so breakdown of bone and release of the calcium, decreased loss of calcium in the urine, increased production of this active form of vitamin D, which will then cause us to absorb more calcium from our gut. Okay? So these three functions, it's acting in three primary sites. In the kidney, it's increasing calcium resorption. It's also increasing the activity of this enzyme which produces the bioactive form of vitamin D. We'll come back to this. This is 25-hydroxy vitamin D1 alpha hydroxylase. And we'll come back to that when we walk through vitamin D metabolism. And that's how it's increasing, having that indirect effect on the GI tract because it increases the production of this bioactive form of vitamin D. And then it functions in the bone to increase resorption and release of calcium. Is that all clear? Okay, so calcitonin is another small protein hormone. It's secreted by the thyroid gland. And it's thought to moderate the effects of parathyroid hormone as well as the active form of vitamin D. It's secreted when there are high levels, high concentrations of this bioactive vitamin D and high concentrations of calcium in the plasma. But it's not clear if this is a major contributor. I think most of the research indicates that most of the absorption is regulated by changes in vitamin D and increases or decreases in circulating parathyroid hormone. So if you completely get rid of calcitonin release, it doesn't have major effects on calcium homeostasis in our plasma. However, it's interesting in that this is being pursued therapeutically to control conditions in which you have hypercalcemia in the blood. So there are many companies that are producing recombinant calcitonin, and then this can be introduced in the blood to decrease levels of calcium. And this seems to be quite effective. So it's not clear that our body is using this as a primary strategy for regulating calcium homeostasis, but it's definitely effective and can be sort of co-opted for therapeutic benefit. So calcitonin kind of does the opposite of what parathyroid hormone does. It reduces the osteolytic activity, so the breakdown of bone. It reduces the formation of new osteoclasts, which are the cell type that breaks down and resorbs bone. And it increases osteoblast activity, which is cell type that builds up bone. Okay, so this is having the opposite effect of parathyroid hormone on bone resorption and buildup. Okay. So any questions on calcium, phosphorus, and parathyroid hormone? Pretty clear. Let's move on to vitamin D. So vitamin D is also known as calciferol. It comes nutritionally in two relevant forms. One is referred to as vitamin D2 or ergocalciferol, and this is produced by fungi like yeast as well as invertebrates, and also from some plants. And this is, its name, ergocalciferol, is derived from ergosterol. It's derived from ergosterol, which is a relative of cholesterol. And in addition, what's really unique about vitamin D is that we can synthesize this in our skin if we are exposed to sunlight. And so in this case, we call this cholecalciferol, and that's because it's derived from cholesterol. And so you can see that these vitamin 2 and vitamin D3 are very highly related. They just have this difference in this side chain here, these slight differences. But they're both available as supplements, and both seem equivalent in terms of rescuing vitamin D deficiency, and both can serve equivalently in a bioactive form in our body. And you can see that these are highly related to cholesterol. If you recall, cholesterol would have an additional ring structure present here, but these are very closely related to cholesterol, which is not surprising that this would be a fat-soluble vitamin. Okay, so in terms of the synthesis of vitamin D in the skin, we're able to take 7-dehydrocholesterol, which is an intermediate in cholesterol synthesis. So cholesterol synthesis is a really long synthetic pathway, but this is right at the end step. So this guy is almost cholesterol. Just has to undergo like one more reduction step, and then it would be cholesterol. So this is right before the immediate metabolite precursor to cholesterol. So in our skin, in response to ultraviolet B photons, this 7-dehydrocholesterol is actually able to absorb these photons, and it undergoes this rearrangement, and it's converted to vitamin D3. You can see this break in this ring structure present here. Then in response to heat, and this isn't like 100 degrees Celsius heat, it's just our body heat, it'll undergo isomerization into this vitamin D3 molecule, which we saw in the last slide. So the two steps are just this absorption of this UVB photon, and it causes this alteration in the structure, forming pre-vitamin D3, and then this isomerization of some of these double bonds to give rise to our vitamin D3. And this form of the molecule can then translocate from the skin cell into the blood and begin circulating in the blood. It complexes with a vitamin D binding protein and can circulate to tissues in our blood. Okay, so our other source of vitamin D is going to be through dietary intake. Okay, so we have one source, which is direct synthesis in our skin. The other source is going to be through our diet. Again, this can come from things like fish, fish oil, liver, egg yolk, or these days from fortified foods. So a lot of dairy products contain fortified with vitamin D. Things like orange juice is a more recent fortification with vitamin D or with calcium, right? So calcium and vitamin D. And a lot of cereals are also fortified with vitamin D. And so this is, not surprisingly, given that it's derived from cholesterol or ergosterol, this is a fat-soluble vitamin, so it's going to be absorbed in our gut through the route of other lipids. So it's going to be incorporated into mixed micelles, it's going to cross the undisturbed water layer, get into our cells, and then once it's present, it's going to be transported via chylomicrons, and then it's going to be excreted from our enterocytes and circulate through our blood, right? And so some amount can be released in adipose tissue, so chylomicrons will have lipoprotein lipase breaking down the triacylglycerol of the chylomicrons, and some amount will be of the vitamin can be released into adipose, and it can actually be sequestered here where it's not readily accessible, and this can be a problem. So it's been observed that obese individuals might actually require higher amounts of vitamin D intake in order to obtain the appropriate serum concentrations because of this propensity of vitamin D to be sequestered to some extent in adipose tissue and fat. Okay, so we have two sources of intake. We can either synthesize directly in our skin or we can intake through our diet, right? The skin, once it gets in, it's going to be circulating in our blood in bound to vitamin D binding protein that's coming through our diet. It will be present in chylomicrons. These will eventually be absorbed by the liver, so the chylomicrons will be in the form of chylomicron remnants. And we'll have delivery of our vitamin D, either vitamin D3 or D2, into the liver. And there are going to be two metabolic events that act on vitamin D to form that active hormone form of vitamin D. And these are two hydroxylation events. One is going to happen here in the liver and the other in the kidney. Okay, so this first hydroxylation event is mediated by this protein called hepatic 25 hydroxylase, or CYP2R1. Okay, and this is going to do what it sounds like, 25 hydroxylase. It's going to hydroxylate the 25th carbon in this molecule. Okay? So we have addition of one hydroxyl group. And this is really an efficient reaction and is not regulated. So this, the amount of 25 hydroxy vitamin D is going to correlate simply with the amount of vitamin D intake, either from our synthesizing our skin or through our gut. So this form, 25-hydroxy-D, can then be released from the liver, and it's going to diffuse in circulation in complex with that vitamin D binding protein, vitamin D BP. Okay? It's then taken up into the kidney where it's going to undergo that second hydroxylation reaction. Okay, so it's taken up into the kidney, and then we have 25 hydroxy vitamin D, 1 alpha hydroxylase. And so this enzyme is what's going to convert vitamin D into that bioactive form. Okay, so just like its name sounds like, it's a 1 alpha hydroxylase. It's going to hydroxylate the 1 carbon in this molecule. So we've gotten addition of 2 hydroxyl groups to vitamin D, 1 in the liver and 1 in the kidney. Okay, so this form, 125 hydroxy vitamin D, can then exit the kidney and circulates again in complex with the vitamin D binding protein. And if you recall, I mentioned that this parathyroid hormone is stimulating intestinal absorption of calcium via vitamin D, this active form of vitamin D. And it does this by stimulating this enzyme. So parathyroid hormone's mechanism of action in terms of uptake of calcium from the intestine is to stimulate this 1 alpha hydroxylase present in the kidney and form more of this 125 dihydroxy vitamin D. Okay, so just an illustration and a summary. We have 2 sources of vitamin D in our body. One is going to be from the skin, right, in which in response to absorption of UVB radiation, we're going to synthesize vitamin D3, cholecalciferol, from this cholesterol intermediate, 7-dehydrocholesterol. The other source is going to be from fortified foods or from other foods in our diet which produce ergocalciferol, which is referred to as vitamin D2. These then undergo 2 hydroxylation reactions, one that's non-regulated in the liver, so forming 25 hydroxy vitamin D, and the second in the kidney forming the, which is regulated by parathyroid hormone, and forms this 125 dihydroxy vitamin D. And this is going to be the active form, which is going to actually have some effects in our body. Any questions on the metabolism? Okay, let's take a quick break before talking about the biological functions of vitamin D. Let's move from talking about the metabolism of vitamin D to talking about its biological function. So as I mentioned, this form of vitamin D, 125 hydroxy vitamin D, is the biologically active hormonal form of vitamin D. Okay, so it requires these two hydroxylation steps. And this is going to be what has the key function, plays a key function in maintaining plasma calcium concentrations. And vitamin D likely plays several other roles in addition to this role in regulating calcium. But calcium supplementation can mostly rescue a vitamin D deficiency. So it's likely that most of its most important roles and functions in the body are in this control of plasma calcium concentrations. So its key effect is increasing intestinal calcium phosphorus absorption. Okay, so as I mentioned, under basal conditions, we're only absorbing about 30%. In response to this active form of vitamin D, this is hiked up to 70–80% absorption. So this is going to be a really important function of vitamin D that controls those plasma calcium levels. It also, similar to parathyroid hormone, has effects on stimulating reabsorption of calcium in the kidney, again, so kind of blocking excretion of calcium. And similar to parathyroid hormone, it also can regulate the resorption of bone and the release of calcium from the bone. So it can induce the formation of osteoclasts, similar to parathyroid hormone. And if calcium is present, it can also function in promoting bone building. So it has these kind of contradictory effects depending on the presence of calcium. So it can induce osteoblast bone remodeling, so either the release or the buildup of bone. And then it actually induces the expression of proteins involved in mineralization. Okay, so if calcium is present, it's going to promote mineralization and the buildup of the bone. And it does this by acting as a nuclear hormone receptor. And so this is a really classic kind of pathway that a few different compounds use. So we can imagine we have vitamin 125 hydroxy vitamin D out and circulating through the body in complex with vitamin D binding protein. Right? It's going to be absorbed into target cells, and then once it's present in the cells, here we see sort of this D3 representing our active vitamin D, has a few different routes. It can either be metabolized, so it can be broken down, and it uses this 25 hydroxylase pathway, or it can be complex with the vitamin D receptor. Okay. The vitamin D receptor is usually present in high amounts in the nucleus, which is this section of the cell, but shuttles back and forth between the cytosol. In complex with vitamin D, it then is phosphorylated. Okay, this is just represented by these phosphate groups. And this phosphorylated vitamin D receptor enhances the association of vitamin D receptor with RXR, which is a 9-cis retinoid X receptor. Okay, and this heterodimeric receptor is an active transcription factor. Okay. So we have the vitamin D receptor. It's bound to a ligand, which is vitamin D. That's 125 hydroxy vitamin D. It's phosphorylated, and then it couples with this RXR to form the active transcription factor. Make sense? And then it binds to these vitamin D response elements in DNA. I want to make sure that people don't get confused that iron response elements are in mRNA, right? Those hairpin loops. Vitamin D response elements, VDREs, are in DNA. Okay, and this is going to affect transcription from the DNA, and it has an effect on a wide number of genes. We won't go through all of these, but you can see that several of these are involved in calcium. So it upregulates a calcium transporter, which is going to be important for absorption of calcium. It upregulates a calcium binding protein, also important for the absorption. This is why it's having those effects in the intestine on the absorption, is by regulating these calcium binding and calcium transporter proteins. It's also upregulating proteins involved in bone mineralization, so osteocalcin. We mentioned briefly when we talked about vitamin K is actually a protein that's carboxylated that doesn't appear to be involved in blood coagulation. Right? And the carboxylation of osteocalcin seemed to allow it to bind better to calcium and to hydroxyapatite. Similar role for osteopontin, another structural component of bone, and alkaline phosphatase also plays a role in liberating phosphate that can then be packaged into hydroxyapatite for this bone mineralization. So it's increasing calcium binding proteins and transporters, it's increasing proteins involved in mineralization of bone. And also interestingly, it upregulates this 24-hydroxylase, which was present in the degradation cascade. Okay, and so this is classic negative feedback. Right? So we have this vitamin D, it comes in here, it has this reaction, it upregulates things, and one of these things that's upregulating is something involved in the degradation of this active form of vitamin D. So this is going to shut down the response, right? It's going to turn off this action. So negative response. So just a summary of these, the important things to take away from this mechanism of action. So our active form, 125-hydroxy vitamin D, mediating all the effects. By binding to vitamin D receptor, this is mostly localized in the nucleus. The key event is upon binding this 125-hydroxy vitamin D, that's the ligand, it's going to induce phosphorylation of the receptor, which enables it to bind this RXR, so 9-cis retinoid X receptor. And this comes in a few different flavors, alpha, beta, and gamma. Okay, so it's forming the active heterodimer. So ligand binding, phosphorylation, heterodimerization, active transcription factor. So this activated heterodimer can then bind to these particular sequences in DNA called VDREs and regulate gene expression. Many of those are involved in calcium absorption or in bone mineralization. So in conditions of vitamin D deficiency, these proteins and these genes are no longer going to be regulated in response. And in response, there's a drop in plasma calcium levels and a decrease in bone mineralization. So that's a vitamin D deficiency causes secondary calcium deficiency, right? So low vitamin D, you're going to have decreased calcium absorption from the GI tract, decreased retention in the kidney, and decreased mineralization. Okay, so in what forms are vitamin D stored in the plasma and retained in the plasma? So conversion, this first step, the conversion to 25-hydroxy vitamin D is not highly regulated. This is just going to correlate with the amount of vitamin D in the body. And so this is going to be readily converted and circulate in complex with vitamin D. So this is going to be in equilibrium with the free form and be readily converted in the liver. And then it can be converted into this form in the kidney, but that's going to be tightly regulated by the parathyroid hormone. Okay, so these concentrations are going to be proportional to total vitamin D, and this is often used as a metric for assessing vitamin D levels in the body, right, and determining whether there's a deficiency. So as I noted before, some of the vitamin D can be sequestered in fat, and how much is sequestered in fat is going to increase as you get more fat or increase as you get more vitamin D. Right? And this is going to regulate the amount that's actually sequestered. So if you have a lot of fat and a lot of sequestration of vitamin D, you might need more vitamin D intake to maintain the appropriate serum concentrations. Okay, so we've gone through a couple steps in vitamin D metabolism. So we have D2 or D3. This is hydroxylated. It's hydroxylated again to the active form. And I mentioned this 24-hydroxylase, which can mediate in it the third hydroxylation reaction. We have 3 different hydroxylation reactions. This hydroxylation reaction is going to form a 24-25-hydroxy vitamin D or 1-24-25-hydroxy vitamin D. And the importance of that modification is that it commits it to a degradation pathway, right? Okay, and this is regulated. So we have regulation by our active form of vitamin D. So as I mentioned, it stimulates 24-hydroxylase. It's going to stimulate the degradation of both 25-hydroxyvitamin D as well as this 125 form. That's going to turn off the response. So it's a negative feedback. And it also suppresses this 1-alpha hydroxylase in the kidney. So it's also going to turn off the conversion of 25-hydroxyvitamin D into that active 125-hydroxyvitamin D. Okay, so it's initiating breakdown and blocking conversion to the active form. Right? So it's turning off the response. Parathyroid hormone has the opposite effect. So it's going to upregulate the activity of 1-alpha hydroxylase. So it's going to increase the conversion into the active form of vitamin D. And it's going to decrease vitamin D degradation by tuning down the activity of 24-hydroxylase. Okay, that makes sense. So parathyroid hormone, it's secreted in response to low calcium. It's trying to increase calcium. It's going to want to increase the amount of 125-hydroxyvitamin D. 125-hydroxyvitamin D is having a negative feedback on itself. Right? So bone is actually, we think of it as a very static element in our body, but it's actually a quite dynamic tissue. So it's undergoing constant remodeling, so buildup and breakdown throughout our lifetime. And it has a unique ultrastructure. It contains both spongy bone, so spongy bone is where our bone marrow is, and that would be where our red blood cells are produced, right? Our erythrocytes. And then it has this cortical bone, which is the more dense, compact bone that gives us the hard structure of our bone. And then the cortical bone is organized into these structures called osteons. These are organized, each one is kind of a tubule organized around a central cavity that contains blood vessels and nerves. And these blood vessels are bringing nutrients to the cells within the bone. They're exchanging gases. They're bringing things like vitamin D or parathyroid hormone, right? So we have to have blood vessel innervation of our bone to provide our cells with these nutrients in regulation. Okay, and so this is just kind of an illustration of what that would look like. There are cavities containing bone cells called osteocytes, and then there's layers of mineralized bone going out from these, within these tubular osteon segments of the bone. Okay. This is just what that would look like in a microscope if you chopped a bone across. You can see this sort of circular pattern emanating from that central cavity with the blood vessels and nerves. Okay, and that's repeated throughout. Okay, so bone is composed, so that's just kind of general anatomy of the bone. The bone is composed of collagen fibers, proteins, and a high amount of deposited minerals. This is mainly hydroxyapatite, which is composed of calcium and phosphate. Okay? And so there's continual remodeling of bone. That's something I don't think we always think about, but there's continual degradation of existing bone and synthesis of new bone, and this balances out to maintain our bone mass. And this continual remodeling is important. So it can adjust the strength of our bone. It's of course important during growth when we're actively growing, during adolescence or other stages we're growing bone. It's also important in replacing the replacement of degrading matrix or collagen and proteoglycans that occurs during normal turnover. You can also imagine if during damage, if you break a bone, there's going to be extensive remodeling and deposition of new hydroxyapatite to repair that bone. And there are a few cell types. Some of these came up during the lecture already that contribute to this bone remodeling. There's an osteogenic cell, which is kind of the bone stem cell and can give rise to these various types of bone stem cells. There's the osteoblast. This is the main cell type that is responsive to vitamin D, the 125-hydroxy vitamin D, and also the main cell type that's responsive to parathyroid hormone. And this cell type is involved in the actual building of bone. So it's going to secrete collagen. So collagen is this tubular protein. It forms long fibrils and forms a scaffold or a matrix, which the hydroxyapatite can then build the mineralized portion of bone. Okay, so this secretes both collagen as well as the proteins involved in the mineralization, that osteocalcin and osteopontin, those proteins which bind the hydroxyapatite and help provide that structure. After this osteoblast becomes surrounded by mineralized bone, it differentiates further and is referred to as an osteocyte, which maintains the bone tissue, and this is kind of like an osteoblast trapped in the bone it's built around itself. The osteoclast serves the opposite role, and so this cell type resorbs bone. And this is a multinucleated cell, and it secretes acids that are involved in the breakdown of the hydroxyapatite for the release of calcium and phosphorus. And this is normally we think sometimes this can be a pathogenic or not a good thing for our body to break down the bone, but this, the balance between the osteoblast and the osteoclast is a normal part of physiology. That serves an important function. So this is just a summary of these cell types. So the osteoblasts, they're the bone-forming cells. They secrete the collagen. It's going to form the matrix for the calcification and the hydroxyapatite deposition. This is the cell type in the bone that actually expresses the vitamin D receptor and is responsive to the active vitamin D. The osteocytes are derived from the osteoblasts, and they're surrounded by that calcified matrix. And then the osteoclasts are the cells that are going to break down and resorb bone. They're very similar to macrophages in terms of looking like a phagocytic type of cell type that's secreting this acid. We're not going to go into the secretion of acid, but in many ways it's similar to how our cells in our stomach secrete acid using bicarbonate and the carbonic anhydrase, that same process of pumping out hydrogen. Okay, so in the presence of normal plasma concentration, this vitamin D can stimulate bone growth. It serves an important function. It does this by promoting the mineralization and the matrix structure, the formation of this prior to mineralization. And it can do this in a few different ways. One is by maintaining the levels of calcium and phosphorus in the plasma at supersaturated levels in this bone fluid. Okay. So it's important for maintaining the calcium concentrations. And then the other role, which we already mentioned, is that it stimulates the expression of these osteogenic osteoblast proteins, including osteocalcin, which is carboxylated in a vitamin K dependent manner, osteopontin, and alkaline phosphatase. Okay? So these proteins are binding hydroxyapatite and providing structure, and this protein is freeing up inorganic phosphate for deposition in this mineral. Okay, so parathyroid hormone is going to influence the release of calcium from phosphorus in two phases. There's a rapid phase in which parathyroid hormone directly stimulates osteolysis, so breakdown of the bone. So this is going to release calcium and phosphate from bone mineral crystals into the fluid around the bone, and then this can be taken up by the blood. And then there's a slower phase in which parathyroid hormone, through its stimulation of vitamin D, is going to promote resorption. So this can actually activate the osteoclasts. It doesn't do this directly, but through its activation of osteoblasts, it presents receptors that control osteoclasts. So it can activate osteoclasts indirectly, resulting in bone resorption. Can stimulate the formation of new osteoclasts from the stem cell niche. And then there's this transient depression in osteoblast activity. And this is all revolving around the importance of parathyroid hormone in maintaining those serum calcium levels. Okay, so we have a few different ideas here. We have the bone matrix, we have the bone mineral, and we have the ratio between the two, the ratio between the mineral and this matrix. So under healthy conditions, these are optimal or normal. Under conditions of disease, these become aberrant. So in rickets, or osteomalacia, we have reduced bone mineral deposition, so we have reduced calcification, right? But the actual bone matrix is still present, so we still have the chondrocytes producing the collagen, and there's a reduced ratio between this mineral and matrix. In there are conditions of osteoporosis, this is where you're actually getting porous bones. Both are going to be reduced, but the ratio between the two is similar. So osteoporosis, this is a really striking image of what this looks like. So osteoporosis just refers to porous bone, so it's a really easy one to remember from its name. And this is really can result from several different conditions, but it's an imbalance between bone resorption and bone formation. And you get this porous bone. And the bones can be incredibly thin and brittle to the point where you're like stepping off a curb and you'll fracture a bone. And you can see this is really amazing. And one of the biggest risk factors is a lack of estrogen, which happens in menopause for women during aging. So this is just a kind of representation of this in terms of the bone mass percent max. During aging, we have this rapid increase as we're growing, and then as we age, there's a gradual loss in bone mass. This is a normal part of aging. But at menopause, females there's a progressive decline at this point and when they stop producing estrogen. And it appears like in females, estrogen regulates the expression of osteoprotegrin, which is an inhibitor of osteoclasts. So in women, when they lose that estrogen stimulation and the release of this osteoprotegrin, there's an increase in bone resorption. So we've talked about osteomalacia and rickets. It's resulting from an impaired matrix formation and impaired mineralization. And this can be caused by vitamin D deficiency, low calcium levels. This is kind of a downward spiral because these conditions increase parathyroid hormone, which is going to increase the breakdown of bone to protect the serum calcium levels, right? So I just wanted to mention that vitamin D has been associated with many other different conditions. It's very clearly, and we understand the mechanisms in quite a bit of detail, regulating bone growth and turnover. But it's also been coupled to cancer reduction, so both breast and prostate. The mechanisms by which it does this aren't precisely clear at this point, though the epidemiological studies seem to support this. It's also involved in seasonal affective disorder, which is a form of depression when you're not getting enough sunlight, which kind of makes sense. And then it's also been implicated in psoriasis, which is often an autoimmune hyperproliferative skin disorder. And this is a synthetic form of vitamin D called Jovenex, which is prescribed and is a topical therapeutic for the treatment of psoriasis. And this seems to be quite effective. And it's, again, not exactly clear the mechanisms here, but it's probably functioning by regulating autoimmunity. Yes, I'll just end here. And we have one more lecture on Wednesday on vitamin A. Feel free to come up and ask